featuring 3 leaders of the FDA. They will share an FDA perspectives on best practice for comparative analysis, challenges, and opportunities. The FDA speakers for this co-lead presentation are Katherine B. Feibus, Commander Andrew Fine, and Jason Flint. Katherine B. Feibus serves as a lead physician in the Division of Device Evaluation Team within the Division of Therapeutic Performance 1 at the Office of Research Standard. Commander Andrew Fine is a senior advisor in the Division of Clinical Review within the Office of Safety and Clinical Evaluation at the Office of Generic Drugs. Jason Flint is a Deputy Director of Division of Medication Error Prevention and Analysis 1 within the Office of Medication Error Prevention and Risk Management at the Office of Surveillance and Epidemiology. The second half will hear insights from the industry and academia. So our industry speaker is Carrie O'Donnell. She is a principal device engineer of the combination product and device research and development from the Teva Pharmaceuticals. She will discuss perspective on risk management and the evaluation of use errors to prevent compromised medical care with generic drug device combination product. Following Carrie's presentation, we'll have a joint presentation from Megan Conrad and Beth Privitera. Megan Conrad is an associate professor of the mechanical engineering at the University of Detroit Mercy. Beth Privitera is a professor of the biomedical engineering and a co-founder of the Medical Device Innovation and Entrepreneurship Program at the University of Cincinnati. They will provide valuable insight from the perspective of human factor experts on determining other design differences and ways to support generic substitutability. So to learn more about our speakers, please visit the CRCG website or you can scan the QR code on the table. So without further delay, I now invite our first speaker, Katherine B. Feibus, to commence our afternoon session. Thank you.
So good afternoon. 3 of us are up here together because we really do all of this together every day. We're going to talk to you about best practices for comparative analyses, challenges, and opportunities. And we're going to see if I can get this to work. There it is. So this is our standard disclaimer that this presentation reflects the views of the authors and should not be construed to represent FDA's views or policies. And what we'd like to do in our talk today is talk about the comparative analyses process and the key groups who really participate in reviewing your submissions when you send them to us. We're going to emphasize some foundational principles and best practices. And we're also going to address other design difference challenges and when a comparative use human factors study may not be required. We'd also like to highlight some of the comparative analyses challenges and strategies when the reference listed drug is discontinued. We know this has been touched on to some degree, but we'll lend our perspective to that as well. So let's take a look at who is doing what when. So the comparative use interface assessment process is extremely collaborative throughout the ANDA life cycle. So you see that there are 4 blue boxes here, or trapezoids on their sides. And we have the OGD Division of Therapeutic Performance 1, which is where the device evaluation team is located. And we are the lead for pre-ANDA comparative analyses assessments. The next box over is also an OGD, which is the Division of Clinical Review. And they actually sit in a different sub-office than we do, but we work with them very closely because they are the lead for ANDA and post-approval comparative analyses assessments. And so we work very hard to talk to each other and check things with each other to make sure that the decisions that we're making and the feedback we're providing are consistent with previous advice. The third trapezoidal shape over is in a different office, the Office of Surveillance and Epidemiology, which is a different super office within CEDAR. And within that super office is the Division of Medication Error Prevention and Analysis 1 and 2. And these two divisions lead review of comparative use human factor study protocols, as well as leading review of results when they come in. Last, but definitely not least on the far right is the Office of Biostatistics. We work regularly with the Division of Biometrics 8. This division works very closely with all of the groups in the Office of Generic Drugs on statistical matters. And so when it comes to looking at the statistical analysis plan and statistical interpretation of results, we really rely on their expertise. And they are in a totally different super office. So again, this is a very collaborative, activity, and we work very hard to make sure that we're all getting on the same page. So I'd like to focus a little more deeply on the pre-ANDA review process for comparative analyses. And you're seeing all of these funny pictures because we don't just read your reports and your submissions. The reviewers on my team do their own comparative analyses and see how their outcomes compare to yours. And so here you can see a picture of one of my reviewers measuring the size of a dose selector knob or dose button. We have actually photo box to take photos, and we have a scale to be able to weigh various devices or pieces of devices. So this is sort of what our process looks like. The comparative analyses process really starts with whether or not a submission is sufficient for providing a device evaluation review. So we look for specific user interface questions being submitted. We check to see that all three of the analyses that comprise comparative analyses are present. We make sure that samples of both the reference listed drug and the proposed generic have been submitted. And if we're missing any one of these things, we will send an information request to make sure that we have all the essential pieces to perform our review. The next piece is reviewing and performing the comparative analyses between the RLD and the generic device user interfaces. And we take a thorough look at the firm's submitted comparative analyses. And then the reviewer on the device evaluation team performs an independent comparative analysis and then compares the findings between the two. We document all of that in a review. We draft up our responses, and we take a look at whether any of our findings are something we've never seen before. And if it's something that we've never seen before, then we will reach out to our colleagues in the Division of Clinical Review and check and see whether they've already reviewed a similar user interface difference in an ANDA in the past that's already been acted on, and try to align on how we interpret this particular difference. Is it minor? Is it other? Is it another difference, but one that has been justified and found to be acceptable before? And so we involve the Division of Clinical Review, and if we need to, we will also reach out to the Division of Medication Error Prevention and Analysis, or DMEPA, to make sure that we are not providing advice in the pre-ANDA space that is at odds with other similar situations. And then the final responses are sent back to the submitter. As you all know, these review timelines may be 60 days. They may be 120 days, depending on whether these questions came in through a CC or whether they came in through a pre-ANDA meeting along with other questions from other disciplines. This is what the comparative use human factor study protocol process looks like in the pre-ANDA space. So it starts on the upper left, and you can see that when a CUHF study protocol is submitted to OGD in a controlled correspondence or a pre-ANDA meeting request, the first thing that happens is it comes to my team. It comes to the device evaluation team. And as soon as we see that, The first thing we do is generate consults immediately out to both the Division of Biometrics 8 and to DMEPA, because they have their own review timelines. We want to make sure that we meet our GDUFA goal dates. And so the first thing we need to do is figure out if we need to engage these other parties. So those consults will be sent out. We also inform our project manager that we are now converting this controlled correspondence, if it is a controlled correspondence, to a level 2. So whenever a CUHF study protocol comes in, that review is always going to take 120 days, in part because of this collaborative process that's needed. So once this is consulted out, the reviewer on the device evaluation team will start putting together sort of the framework for our review that will ultimately house the response that will be sent. But in the meantime, DMEPA and the Division of Biometrics 8 are doing their reviews from their human factors and statistical perspectives. They communicate with each other. When we send these consults, we let them know that the other division has also been consulted so that they will communicate with each other as they're reviewing the protocol. And ultimately, they write their own review and recommendations, which we then incorporate into our final review before the response is sent to the firm. So this is how we coordinate this process. I'm now going to hand this over to Andrew, who's now going to show you how this process looks in the ANDA review stage.
Thank you, Kate. Good afternoon, everybody. My name is Andrew Fine. I'm the Senior Advisor in the Division of Clinical Review. And as Kate mentioned, the Division of Clinical Review is the lead for the comparative analysis assessment during the ANDA phase as well as the post-approval phase. And really the importance of walking through, some of these confusing flowcharts is really with our industry colleagues here, knowing kind of the players involved and when they're involved and when you may be receiving communications and kind of the aspects of the back and forth really just help the evolution of these applications and allowing timely feedback and timely response to questions that are posed to industry. So I'll start with the CA review process, and I'm using the original ANDAs as my case here because this represents about 85% of this comparative analysis work in our division. When that original ANDA is submitted, like every ANDA, it is assessed first by the Division of Filing Review. And for these types of reviews, what's really important is to identify early whether that ANDA is a drug device combination product. So that takes place within the Division of Filing Review within the first 2 weeks of ANDA receipts. If a new ANDA is identified as a drug device combination product, a DCR review team is assigned within a few weeks after that. And therefore, our review team can quickly identify and triage that application and ask any follow-up and prepare an immediate information request, whether we need a more complete or a full CA report, whether we're requesting product samples or additional pictures, really to guide our assessment. Once we have the information we need, DCR completes their assessment by that mid-cycle date. For those involved with the evolution of GDUFA and GDUFA 3, this paradigm here is representative of the other disciplines within the generic program, where that early communication, that initial communication is sent around the mid-cycle date for all these original applications. So as a result of our comparative analysis, if we have existing deficiencies as a result of that assessment, labeling deficiencies or device deficiencies will be communicated via an IR, but behind the scenes, DCR is coordinating with our labeling colleagues, with quality colleagues, because any questions we have or requests for changes as a part of our comparative analysis will affect those sister disciplines in labeling and quality because there's a shared responsibility for that application. Following, if deficiencies are sent around the mid-cycle, the team will review that applicant response and determine whether that assessment is adequate or if there are any remaining deficiencies, those would be communicated in a complete response letter. Another subset of this process is the work we collaborate with DMEPA. I'll talk about in the next slide about the comparative use human factor studies, but if there are questions about interpreting the comparative analysis assessment or understanding the user interface for a proposed product, we also may involve DMEPA to help collaborate on that assessment. But if we shift to those situations where an original ANDA It also includes a comparative use human factor study. The initial steps are the same in terms of assignment to DCR, but because of the expertise in DMEPA and the biostats expertise and the stats group, following receipt of that application and determining that initial triage, which includes those samples or other requests, we immediately consulted our colleagues to help assist with the assessment of the comparative use human factor study and the statistical approach in that study. Those consultants may then submit additional questions about the assessment and send IRs themselves to the applicant to guide their review. Once the assessment of the comparative use human factor study is done, there's a multidisciplinary discussion amongst DCR as well as the consultants to determine an outcome for that review. And then it'll be either adequate or we would send that complete response letter. So this session and a lot of the workshops today are going to be about other design differences and really collaborating on how we can work together to navigate methods and alternatives and how we can approach some of these user interface spaces that are inevitable based on a lot of the discussion this morning. But I really want to take a step back and really reinforce and highlight a lot of these foundational principles for user interface, for comparative analyses, and some best practices that really can kind of guide us as we have these tough discussions. So this was discussed before, but remember, therapeutic equivalence is the mainstay for our regulatory framework and generics. And I'm really highlighting the key points of these regulatory principles when it comes to the user interface assessment, and that is really that generic product, including drug device combination products, are expected to have the same clinical effect and safety profile when administered to patients under the conditions specified in labeling. And we consider whether end users can use that generic combination product without the intervention of a healthcare professional or additional training. And we know that it's challenging to make these products identical, and they do not have to be identical. But any differences should not preclude their approval under an ANDA, and really tying this back to the therapeutic equivalence, those differences should not impact the ability to have the same clinical effect or safety profile as the RLD. I don't think this guidance is unfamiliar to anyone in this room, but we have to have the obligatory slide for comparative analysis guidance. There are three comparisons: the labeling comparison, the comparative task analysis, and the physical comparison. And we'll talk about how we are implementing these, how we do these as we approach our comparative analyses. So these definitions may be familiar, they probably are familiar, but this is the foundation of a lot of the principles and how we apply these comparative analyses. So I do want to take a minute just to reinforce what these definitions are. First, the user interface. So these are all components of the product for which a user interacts. It includes the delivery device constituent part, any associated controls, displays, labeling, and packaging. Critical task. It is a user task that, if performed incorrectly or not performed at all, would or could cause harm to the patient, or that harm is defined to include compromised care. And then the external critical design attribute is a feature that directly affects how users perform a critical task in order to admit it to use or administer the product. Our comparative analysis outcomes. So for each difference identified, it would either be no difference, minor difference, or other design difference. And the distinction between the minor and other design differences is whether that difference may impact an external critical design attribute. So going back, knowing the definition of critical task, using that to identify critical tasks allows appropriate classification of these differences and justification. But all that assessment must consider not just the differences themselves, but the context of use and the risk profile of the product. The regulatory framework, we use therapeutic equivalence, but we apply that individually to each RLD. The RLD will dictate that risk assessment. So taking this into some overarching best practices that we'll dive into detail throughout this day, I'm sure. So first, identify all user interface differences. In a comparative analysis report, if you know there's a difference and don't identify it, we will identify it. Classify all those differences, but use the definitions and the guidance. We're talking about other design differences. If you think something is other design difference, call it another design difference and explain why. And that allows you to focus on those differences in critical tasks. And remember, the definition of a critical task does not apply to all user tasks. So we have to keep in mind that not every task is a critical task. And again, consider the product and its context of use, focusing on that individual RLD. The same difference may be classified differently based on the RLD because the context of use, the use environment may change across those RLDs. So in terms of context of use or use environment or environment of use, we've defined this and broken this down into a few areas that help us understand the RLD and the impact of any difference. So there's urgency of use. Some products may be used more urgently than others. Some products are for a single use versus chronic use. Who is that end user? It's very different if the end user is a patient or patient caregiver versus a healthcare provider. Where is this product used? Is it used in a school, in a home, or in a hospital or clinic setting? And knowing the patient population is important as well. The patient population may have difficulty using a certain device design because of underlying disease state, dexterity challenges. This should all be incorporated into that assessment of each of these differences and classifications of those differences. So with that foundation, I'll turn it over to Jason, who will dive a little bit deeper into these other design differences.
Thanks, Andrew. So let's talk about these other design differences. I'm going to try and sort of do a little bit of myth busting, hopefully. So Andrew presented these terms before: critical task, critical design attributes. And so just to review, critical task is a user task that if performed incorrectly or not performed at all, would or could cause harm to the patient or user, where harm is defined to include compromised care. And so the external critical design attribute definition incorporates that critical task definition. So those are features that directly affect how users perform a critical task. The attributes of the product would be those features that end users rely on to safely and effectively perform critical tasks. And we hear this a lot, so I'm going to ask the question, does this mean that every task that could result in a medication error is a critical task? And it's from the audience. And that therefore, all other design differences, all differences are other design differences? No. Okay, good. No. So what then should I consider when I'm identifying a critical task? One is the potential for harm. One is whether the task impacts directly administration or dosing. But another might be urgency or time, or is urgency or time. And so a particular task for one product may be a critical task or not a critical task, depending on whether that particular task relates to harm or relates to sort of delay in application when urgency is a problem. So consider something as simple as cap removal, right? If I have a denosumab product, right, so that's a product that's not for emergency use, would cap removal be a critical task? No. Epinephrine, right? Okay, so we're getting the idea. Not every task is a critical task. You have to consider the potential for harm. You have to consider whether it impacts route of administration. You have to consider whether urgency is or time is of the essence, right? Or whether it has the potential to impact dosing, right? And that's how I'm determining those critical tasks. So one of my favorite slides, right? Oh no, I've identified another difference, right? So take a breath. It's not time to panic. Do I need a CUHF study, right? So this is the other question that we get all the time. If I have other design differences between the user interface of the RLD and my generic, do I have to conduct a CUHF study? No, not necessarily. And we'll get into some examples. So what options do I have? Right? When my comparative analysis identifies other design differences, I could consider redesign. I would argue, based on some of the discussion this morning, right? That I should be considering whether I'm developing something that inherently has another design difference, maybe before I start going down that design pathway. But we'll put that aside. So I could consider redesign of that proposed generic user interface to minimize difference from the RLD, right? Again, I would probably consider that from the design side, maybe first. But you might need some additional information or data to support your ANDA submission. And by the way, you may already have that information that we need. So let's work, what else can I do, right? I would want to work through that scenario. Okay, what happens if a user engages that other design difference inappropriately? What's the risk associated if that anticipated error occurs? Does it impact your proposed generic having the same clinical effect and safety profile as the RLD? Sometimes that answer is going to be no. But tell me why or why not? And then what other things can I do to address this? Right? So is there other information that I might be able to use to sort of fill that data gap? All of these answers are going to be affected by the drug product profile, the intended users, the use environments, the context of use in general. Okay. So let's get into some examples. Example one is a dry powder inhaler. There's a product-specific guidance for fluticasone propionate and salmeterol xenophoate. Apologies, I'm not a pharmacist. Posted in September 2013. The recommendation includes studies of bioequivalence, formulation sameness, and device considerations. This was the first generic for that DPI approved in January 2019. And there were some device and user interface considerations along with that approval. There were other differences addressed with additional data in that ANDA. So here is a picture. This is Advair on the left, and the generic on the right. And so the difference here is probably the most important one, or the one that we'll discuss, is the orientation of the inhaler itself. The Advair inhaler is held like a hamburger against your mouth, sort of parallel to the ground. The proposed generic is held in a sort of a taco configuration, right, perpendicular to the ground. I don't have examples, right, but now you'll remember that. Okay. So the question then became, well, okay, is that an other design difference? Anyone? Yes, it is. Okay. So the question then became, well, the inhalation steps, maybe those are the same, right? What happens if I have this orientation difference? The potential use error is following generic substitution. The user could hold that DPI in the horizontal position, like the RLD, rather than the intended vertical orientation. Okay. The potential harm associated with that is that it might alter the drug dose reaching the distal airways, and therefore alter overall efficacy and safety profile of the generic compared to the RLD. Okay. Do I need a CUHF study? That could be one way. In other words, I want to show that my users aren't making that error, right? They recognize the difference, they're holding it in the right orientation. That might be one way. In this case, though, the applicant submitted data demonstrating that even if users made that error, it didn't affect safety, right? It didn't change the clinical profile, right? Didn't affect the drug deposition pattern. Okay. So example two is one that comes up quite frequently. So this is a presentation where the RLD comes in as a vial and the proposed product comes in as a pre-filled syringe. Clearly, there's another design difference, multiple, right? But when is this an acceptable other design difference? When do I need additional information or data to support the difference in presentation? So some things you want to consider. What's the context of use? What's the environment? Who are the intended users? What are the labeled indications in dosing? And what's the risk associated if errors occur? If it's a situation where it's healthcare providers and I'm delivering the entire contents, We have to consider, have I introduced risk? Have I done some other things, right? You might get a very different outcome than if I'm considering that this is for lay use, right? And I have to adjust dosing and I have to do these types of things. Again, going back to that slide about what's the harm, right? Does it impact administration? Does it impact the dose, et cetera? So in that situation, we may find that there are acceptable differences in the user interface without additional support from CUHF study data. Things that may impact this decision, this isn't all of them. I encourage you to think of others. Are there critical tasks that have been removed? Right? Generally speaking, that's desirable. But have you introduced a new critical task? For example, would that drug need to be expelled, or would some drug need to be expelled from the prefilled syringe to achieve the correct dose? And has the prefilled syringe been designed to support all the labeled dosing? So does it have the appropriate syringe markings to support, for example? And so I think this is back to Katherine for best practices when the RLD is unavailable.
Thank you. Okay, so we'll go back and revisit a topic that came up a little earlier. So it is no fun trying to figure out how to do your comparative analyses when your RLD is unavailable. And certainly we had a related question that came up during an earlier panel discussion where the example was brought up that one of the Ozempic presentations has been discontinued, one of the variable dose pens. And what do you do? So we don't like this either. It's hard for us to do the comparative analyses without the RLD. Oh, that should have happened. I'm sorry. How do we back up? There we go. Sorry. I thought I'd removed all of the hidden animation that wasn't supposed to be there. So it's hard to do, but you still have to compare your proposed generic against the RLD. You may have to take a somewhat different approach, but you have to figure out how to do it. So while normally it is easiest to start with a physical comparison in a lot of ways with the comparative analyses, when the RLD is unavailable, starting with the labeling comparison can be very handy because often if the RLD is unavailable, often the labeling is available. And so you can find it at drugs at FDA, you may be able to find it somewhere else, and you may be able to at least start putting together the borders of your jigsaw puzzle using that labeling, figuring out exactly how that RLD was supplied, figuring out the different ways it was presented, what the instructions for use were, whether it actually has separate instructions for use or just step-by-step instructions in the dosage and administration section. And that's often a good place to start. And that can then inform putting together the comparative task analysis. The physical comparison is definitely more challenging when no samples are available. And there's a couple of creative approaches you can take to help. So let's just touch base first on the labeling comparison. Again, you can find it at drugs at FDA in many situations, but sometimes there are old drugs that have been discontinued for a long time, and there is nothing. You cannot find the labeling in the public domain. And we know because when that happens, we look and we make sure it's not in the public domain. We want you to have what you need to do these comparative analyses. So if we can locate it and you haven't been able to locate it, we will tell you where we found it. If it is not available anywhere other than within FDA files somewhere, there have been instances where our policy group has agreed that because approved labeling is public information, that we can send you the most recent labeling for the reference listed drug if it cannot be found in the public domain. So Lisa Berku mentioned earlier when she was on the panel, if you cannot find labeling in order to put together your start putting together your comparative analyses with a discontinued reference listed drug, please reach out to us, send us a controlled correspondence, let us know what you're struggling with, and we will make sure it's okay. But if we are able, we will share with you that most recent labeling so that you can begin your process. The physical comparison is more challenging, and it sometimes requires more creativity. So, for example, if you have a situation that Jason was exploring where you're going from a vial to a prefilled syringe for your product, there's not a whole lot to know about a vial. Most vials work exactly the same way. And, anyone who works in human factors for medical devices or any healthcare provider is going to be able to lay out exactly what steps are needed in order to properly draw up drug from a vial, put it into a syringe, prepare the dose, and just walking through those steps mentally, even when you may not have a physical sample of the RLD, can be really helpful for starting to build out that comparative analyses. The other thing that can be really helpful is finding sources for images or sketches, some of which may be available in labeling, and you can certainly use those and rely on those to the greatest degree possible. But sometimes you may have to think about what other drugs are out there that would be good examples of a similar situation. And it is reasonable when you do not have samples of the reference listed drug to say, well, we've done our research and we think that this product or this particular container closure is a really good parallel to this unavailable reference listed drug. And so we do encourage you to try to put together the most thorough comparative analyses you can based on the information that you have available to you, and send it in to us, get some pre-ANDA feedback. And we understand what you can and can't see. We will take a look at what we can see and provide appropriate feedback based on that. So we recognize that this is a challenge and we do struggle with it internally at times as well. So you can see the other suggestions about other sources of information, but we know by the time you've reached out to us and said we can't find anything that you've probably done a really thorough job looking. So for the comparative task analysis, again, you can sometimes build quite a bit of that from labeling. If you don't have that and you're dealing with a fairly simple device, such as a dosing cup or a syringe, how these particular types of devices are used in order to administer a drug and to measure dose can really be deducted just by the way the device is made. So again, you can build out a comparative task analysis based on what is known about how you administer a drug from an oral dosing cup or from a syringe. And it is reasonable to justify building out your task analysis that way to some degree if you need to. There's additional descriptions you may find of particular container closures. Definitely include those and include steps that are relevant to those as needed. And if the proposed container closure is different from the RLD, then those tasks in your task comparison should, of course, reflect those differences. So again, just a reminder, the primary comparison should be against the reference listed drug, even when it's not available. You can use other products, whether it's the reference standard or whether it is another drug that is used in a similar setting by a similar population. You can draw parallels and use that as supportive information to help fill in the empty blanks in your comparative analysis when the RLD is discontinued. The three of us want to say thank you. We appreciate all of you being here, and thanks for listening today. Right.
Good afternoon, everyone. So my name is Carrie O'Donnell from Teva, and my talk today will be on risk management and evaluation of use errors to prevent compromised medical care for drug device combination products. Okay. What am I doing wrong? There we go. All right. As with all the other presentations, the standard disclaimer that the opinions expressed in this presentation are those of myself and not necessarily those of Teva Pharmaceuticals. I'm going to start with an introduction to myself, really to lay the groundwork for where this presentation is going to go. I had the unique opportunity actually to begin my career at a place where typically most people would end their career in forensics. So as soon as I left university, I worked for a forensic engineering firm. My expertise was in injury biomechanics, medical device failures, amusement park ride operation design, crash reconstruction, product liability, and metrology. So this meant that based on my education, training, and experience, I would provide scientific investigations into typically these failures or incidents, where people got injured and would figure out what went wrong. Was it a product issue? Was it a use issue? Was it an environment issue? And because of that, because that was where I started my career, kind of got a trial by fire, and had a process ingrained in me on how I look at products, how I look at use scenarios. And it's really from a standpoint of failure rather than this is a great device and it's going to work and this is how we designed it for the first time. That's how we like to look at things, but rather my vantage point is this is going to fail. How is it going to fail? So that's really the now, since kind of working on the other side and working in product development, that's the point of view that I take into all of my product designs and the view that I'd like to share with you guys today. So what are we going to talk about? We are going to talk about the approach where use errors are identified for critical tasks, for other differences, how the use error could have potentially no impact, that binary endpoint that many presenters have talked about earlier, the risk management approach for clinical impact of use errors over the intended treatment length, negative transfer versus other root causes for use errors associated with other differences, and user learning and repeated use in the context of intended use. So how does the risk management process apply to device user interactions? So we know we have a device, that is the user interface. That is the way that the user is going to interact with our product to eventually get that clinical efficacious dose, hopefully, right? That's the intent. So we have to take into consideration the use environment, the user, and the user interface. And comes out of that, was it correctly used, or did it result in a use error? We also have to take into consideration those hazards, so those sources of harm that users could be faced with when they are using their product. Did we design these in? Is this a product of the use environment, that this is product that's been used in other places, but this particular environment is causing problems? So where do these crop up? And what are these? These are all things that we need to take into consideration throughout the development process. So examples. The device exceeds the ability of the user, the cognitive ability of the user. It's too difficult for them to use. The device's use is inconsistent with the user's expectations. This is something that is critical for us as we're creating generic drugs, right? The use environment affects the operation, whether it's in a hospital setting, at home. The particular use environment impairs the user's physical perception or cognitive capabilities when using the device. Devices are used in ways that the manufacturer could have anticipated but did not consider. And the devices are used in ways that were anticipated but inappropriate. These are two pieces at the end. We'll go into a little bit later but foreseeable or unforeseeable misuse. So we know the ANDA model by statute says that therapeutic equivalents are approved drug products that the FDA has determined are pharmaceutical equivalents for which bioequivalence has been demonstrated and can be expected to have the same clinical effect and safety profile when administered to patients under conditions specified in the labeling. Sorry, I should finish the sentence. So what does this mean for device development of a generic product? This means throughout the development process, so from feasibility all the way through post-market, the benefit risk of any design difference between a generic and an RLD, as well as the root cause of any use errors, should speak to the maintenance of the bioequivalence and pharmacoequivalence of the combination product. So this is where we're getting into those primary endpoints of use here. Are these differences, could these impair the effective use of our combination product? Could it impair the clinical efficacy of the combination product? This should be in the back of our minds through the risk management process throughout the development process. So we have numerous methodologies throughout risk management that we can use to inform our device development, our design. These could be the hazard analysis, failure modes and effects analysis, fault tree analysis. And the outputs of these analytical methods inform our URRA, our use-related risk assessment, which identifies the critical use steps required to meet the clinical endpoint. Like I said, the successfully receiving a clinically efficacious dose. Now I want to pause and caution a little bit. Some questions have come up in earlier talks about when do these activities take place? When should we be doing these exercises? We want to make sure that risk management is not considered a checkbox exercise. And what I mean by that is that it is not a linear process, right? We're not creating our device development plan, and then we go to this step, and then we go to DV, and then we go to human factors. It's not the way it's supposed to work. From the very onset of the device development, prior to that, design phase 0, when we're working in feasibility, we can be doing parts of our use-related risk assessment. We can be, even without having physically acquired the RLD, making assessments, looking at labeling. We can be doing early formative studies that are informing us on our platform technology selections. All of these things are eventually informing the potential risk that we could be bringing into our technology and could be asking our patients to face. Or it could be lowering a risk that we initially thought was of a higher severity, but we have data now to show that it's not something that could be considered another difference, and maybe it's a minor difference. All of these things are iterative. These are green documents, for lack of a better term. So I want to make sure that we all understand that from the very beginning, we can start this stuff. This is not something that we have to wait 2-3 years on the line to do. So there's some examples. So going into, we brought up critical tasks. As I said, these should be assessed on the severity of the potential harm that could result from the use errors. And this is identified in the risk analysis, sorry. Along with this, as we're going through our task and we are looking at the task that we're asking our users to perform, the foreseeable misuse should also be considered. So even though it's not part of, this is the way that we've designed it, this is the way it should be used, we're good to go. We know that's not always the case. And so we need to consider those situations where our device could be used incorrectly. Is that foreseeable? Are our directions not clear enough? Is our design so different that it could cause the user to use it in a way that is not intended? This is foreseeable misuse and does need to be part of our risk management process. So some examples of critical use steps and then how they might relate to our essential performance requirements. For a prefilled syringe, identifying the ejection site, push the plunger to the bottom of the barrel, engage the safety guard. This relates to dose accuracy, break, loose, and glide, and then NSD override. For a pen, prepare your new pen with a needle, select your dose. For both pen and auto-injector, inject your dose. Essential performance requirements, dose accuracy, activation force, and injection time. So all of these use steps also have a related design attribute that we would be considering, again, from the very beginning through our human factors studies and onto market. For a generic, the additional piece that we also have to consider is that part of negative transfer. So where are a potential other difference could relate to confusion, or that could contribute in a way that may affect successful administration of the dose. So root cause analysis would be conducted to determine the impact of negative transfer and how to mitigate that risk to as low as possible, judged against the impact of pharmacoequivalence. Again, negative transfers, if this is something that may be present in your generic device, you can start gathering these data at the very beginning of your device development during formative studies, and you don't need to wait until, say, we get down the line. That we get down the line and you have to perform a comparative use human factor study. You want to be thinking about ways you can reduce your risk throughout the process, whether it's a design difference, et cetera, and where you would be gathering those data to support whatever decision that you make. So my previous co-presenters have talked about this, but as a reminder, things that go into threshold analyses, labeling comparison, comparative task analysis, and then physical comparison. The results of these then could lead to the conclusions of either no difference between your device and the RLD, minor difference, does not affect the external critical design attribute, or another difference when design differences may impact the external design attribute. Other differences, and I know this has been brought up a couple times too, what are ways that we can justify other differences, that justify our risk severity scores for other differences? Something critical that I also want to bring up is that we want to, whether it's a generic drug product or an innovative brand product, we do want to always make sure that we are taking an innovative mindset to development here. We want to be best in class, right? We want to be creating a user experience that is as low risk as possible for the user. So if that means, based on your risk assessment, that you need alternative safety design or just an alternative activation step, these are all part of your risk management process, and areas where you can justify these differences and gather those data to support that is, as what has been talked about, comparative use human factor study, bridging data from other programs, and there is a FDA guidance on that, on bridging and how to use that from either formative studies, or say you have a platform technology in your pipeline, you've used it for another product, you have data on one aspect, you need to gather additional data to fully support it, but you've got the beginning there. Early formative usability studies and biomechanics and related data, such as what was brought up earlier today about post-market studies and how you may use those data in a later study. Those are all examples, certainly not limited to those. So sticking on the topic of other differences, I said, we do want to take an innovative approach to generic development projects. Even though we have something to work off of, we always want to ask ourselves, is this the best that we can be presenting to our patients? Is there something else that we should be doing? So state-of-the-art, best-in-class alternative designs. So as a device safety feature evolves, following the safety hierarchy or hierarchy of controls, as some know it, how might those differences affect the use? Again, could this relate to negative transfer? The principles here inform certain device design elements, and relate to the safety and ease of use, regardless of if it's a generic or an innovative. And then the threshold analyses, risk assessments can help focus these usability studies through the development. Again, from the very early stages when we're still trying to decide if this is the technology we want to use, do we need design differences, through to validation. So identifying those higher risk steps or user subpopulations that may affect successful use. Determination of impact of related use errors associated with a change in external critical design. And then sometimes labeling. If there is a difference, how different do we need to make labeling steps to cover that difference? As we know, labeling for a generic needs to be to match what the RLD is, but then how do we cover those differences in design of the proposed generic that needs to be covered in the labeling? So I want to go over an example of how we might put all these principles together. So we have a proposed generic two-step autoinjector for the drug substance fake drugatide. The RL presentation is not real. It's a four-step autoinjector. So we can see the differences on each side here. Major differences here is the platform AI does not have a button on top. The not real branded AI has the button here and it has an interlock. So typically, oh, sorry about this. The colors seem to have moved a little bit. Typically, what we would see for likely to be an other difference could be the button on the top or lack thereof. Something that could likely be a minor difference is the grip. For the autoinjector, one is hexagonal, one is circular. This could be something that is critical for your user population. If this is something that's going into maybe a rheumatoid arthritis population, and grip is something that could very much affect the proper use. Negative transfer here. User interface, again, between the generic and the RLD that may contribute to use errors, difficulties, and or failure to successfully administer the drug. At the end of this and throughout the setup of your study and at the end of the study when you're gathering, doing any root cause, gathering your conclusions, root cause analysis and risk assessment would need to determine if there was an impact to pharmacoequivalence based on these differences. So how are we considering risk in all of these? The difference between a minor and another difference through alternative approaches. So we could utilize the threshold analysis, the bridging framework to bridge from an other validated device with overlapping use specs to the innovator, potentially. Summative style validation with users susceptible to negative transfer to gain objective evidence of the probability of negative transfer or lack thereof. This could be done during a formative study at the very beginning to judge whether this is going to be a risk for you. The use profile, the recruitment effort, use environment, and then the critical use steps. These are all areas that could be presenting risk into your product, and that may not be the same from product to product, especially if you're using a platform technology. So these are things that can be considered at the very beginning of your development, and you don't need to wait until it's time to do a comparative use human factor study. How do we interpret the results of those areas that may be outside of the critical use steps, but still may contribute to risk to our patient population? How many engaged with the new IFU? We cannot always rely on just information for use. Sometimes there does need to be a design element factored in if there are significant use errors. How many successful injections? Success, again, should speak to the primary endpoint of completing a dose. And was there evidence of negative transfer? What was the use, the root cause of this negative transfer? So, summaries. We should be prioritizing benchmarking performance of potential solutions against state-of-the-art, best-in-class for our device decisions. We should leverage use-related risk management tools starting during feasibility stage, design phase 0. So doing small-scale formative studies to pressure test. And then again, the bridging guidance can be something that can inform how data may be used across programs. Do a threshold analysis early and often. Again, this is a green exercise. It can be changed with new information, and you don't always need the physical samples to anticipate potential differences. Minor and other differences may be supported by analytical means identified through the risk management process. Other differences may still require a comparative use human factor studies with sample sizes scaled to the severity of risk. Review this with the FDA. And then use errors should be assessed against the primary endpoints and determinations made by impact to bioequivalence and pharmacoequivalence. And finally, review your substitutability strategy with the FDA via controlled correspondences and pre-ANDA processes early and often. And then just some light reading for everyone. And that's it.
I'll take this opportunity to introduce myself, and maybe Megan, you can introduce yourself. So hi, everyone. I am Mary Beth Privitera. I am from the University of Cincinnati. I teach biomedical engineering there, and I am co-chair with Molly Story, who's in the audience here on the AMY Human Engineering Committee. So this is all near and dear to my heart, and I'm going to be co-presenting today with Megan. You want to introduce yourself?
I'm Megan Conrad from the University of Detroit Mercy. I'm an associate professor in mechanical engineering with a background in biomedical engineering as well, and working with Mary Beth and Molly and others in Melissa Lemke, Molly Laird, on a project with the Office of Generic Drugs.
Yeah, so thank you all for sponsoring our research, and we're going to share a little bit about that. For those of you that know me, know that I bleed design. So the first part of this conversation is going to be me entertaining you about design, since design is such a primary important part of this generic process. So I'm excited to be here and talk about product design and bring in that perspective. And again, thanks to the FDA for supporting our efforts in this area. Okay, let's see if we get going here. All right, so product design, the first part of this guidance for the CUHF guidance is to talk about product design and how you're going to categorize that, right? Minor versus other. So I thought it would be a great opportunity to just talk about what is product design. My research, even though I teach in biomedical engineering, I'm not an engineer, I'm a designer. So a little bit of different background. And what constitutes product design are appearance, functionality, manufacturability, value, and does it improve life? Now, you can argue that not all products do improve life, like the fish that moved that was out a couple of years ago. There's lots of dumb products. So we don't always have to improve life, but I'm sure that probably did improve someone's life somewhere along the line. But when we talk about product design in general, we're really talking about the overall form, the overall details, and it can be a couple of different things. And so I'm going to blast through just a little bit about some form and just talk about it in general. So the form of an overall design can be something that's geometric. It can be minimal. I think the first minimal design that really hit was the whole iPod. The iPods, when that first came out, that was very minimal in comparison to your 8-track cassette tape. That was my age, right? And then it could also be biomorphic design. It can be soft, it can be flowing. And you can see that sometimes it functions the same way. The chairs that you're sitting in versus the chair that you see here, they're very different. One is very angular. You like some, you have different preferences. You have different ways of interacting with it. So the forms can either be one or the other. They could be both. They could be combinations. In addition, they could also be playful. They could be fun. These are all neo-pop design where they have a playful aesthetic. And they're still functional though. They're still product design. They're still mass produced. They're still products, but they're interesting. And what makes them interesting is the materials. How do we use the materials? How do the materials come together to form that product design? And you can use a myriad of materials, although I do know that most pharmaceutical companies don't like to change the material that the drug is around because that's a whole headache. So we won't talk about that material. We're talking about the material on that user interface. The other part of it is about the finish, and the finish gets to how is that finish related on that material. So it's not just the material itself, because the material itself can be variable in and of itself around the different shape, but how is it finished? Is it shiny? Is it flat? Is it textured? And how do those finishes relate? Because that all tells a user how I'm going to actually interact with the particular product.
And that also includes things that are mechanical features like grills and vents. And sometimes we have to have vents. We have to have features that enable the functionality of the device, but they're ever more important and increasing to the functionality of the device. But if I put it in the wrong spot and my hand blocks it, then the device doesn't actually work the way that I want it to. In addition, it gets down to the buttons and the knobs, and these are all the elements in a product design that are important. The elements and the knobs, the buttons and knobs that you see here, those are all the important parts that we're going to interact with. How do we know that we're going to interact with something? How does that change over time? How do we know to move one button this way or that way? Do we do push? How many people here have gone to a door and they've pushed it when they needed to pull it? Or they've pulled it and they've needed to push it, right? These all communicate things of how we go through our daily life and can be either annoying or something that we love and we enjoy. In addition to screens, we can think about the screens, and screens are becoming ever increasingly important in our world. And that's true of all of our combination products as well. You're going to have app-driven screens. Screens have many, many different uses. The design of the screens, the user interfaces themselves, they can either be hidden, like the little high on the bellow device that you see on the lower left. They can also break up information that you see on the dashboard. So how we handle the screen, it's not just the fact that it's a screen, it's to the information that's on the screen. How do we handle the color that's on the screen? How do we know if that user is going to be using that screen in light and dark? How will it work in those various applications? In addition, graphics and logos. Let me translate. Graphics and logos, that's what we tell of our product design students and our engineering students. But for everyone in this room, it's labeling. So think of this one as labeling. How do we handle our labeling? How do we label our buttons? Where do we label our buttons? Do we need to label our buttons? How about icons and symbols? I know that on the standards committee, there's an international standard on the icons and symbols that we use in medical devices. And I doubt that they were ever really robustly tested in a human factors world because they've never, ever scored very well on any human factors test I've been involved in. But they're important, and they're an important part of our product design. And the one thing I'll point out is that design is always evolutionary. You're always building off of something that came before you. And if I look just simply in one product development process here, you've got something that's functional engineering that goes into product design. And design changes. So here's another example of the evolution. And this is the Mini Cooper, which is also near and dear to my heart. And if I take a look at the first part of the Mini Cooper design in the classic of 1959, we can look that it only measures 3 meters. And then in 2010, it's 4 meters. Are these devices the same? Are these products the same? Would you argue that they're the same? I would argue that if you're driving the 1959 classic Mini Cooper, you really need to like someone because they are literally sitting on top of you in that little car. And then 2010, they're a little bit farther away. So while they do appear to be the same, they've evolved to be something that might be a little bit different. And so I'm going to bring you ketchup. Ketchup. If we can agree that the ketchup bottles configurations that you see here they function the same. They all deliver ketchup, just in different volumes. And since there's no risk of ingesting ketchup, each configuration is inherently substitutable. Do you agree? They're all delivering ketchup. Therefore, it's perfectly acceptable for a gallon of ketchup to be delivered through a drive-through window. While it might be safe, it doesn't meet their user expectations, and it may introduce additional risks. So are these the same? Are these substitutable? It really depends on the task. It really depends on the context. Oops, sorry, I didn't mean to do that. That was a new one. We had a good flow too, so just come on back with me. Obviously, comparative analyses for generics are not as easy as ketchup. They're highly more complex than what I've just shown, and there's a lot more potential harm, a lot more danger that can happen and are substitutable. So we're looking at it closely. The problem with comparing devices and designs is without a common knowledge, without common language that describes the design, things like the materials, things like getting down into the details of how the materials, the forms, the shapes, the functions, the features, without having a common knowledge that we all share, it's hard to describe them. So while we're asked to be going into other or minor, what language are we using to put it into that category? In our research with the FDA at one meeting, I will distinctly remember this meeting forever. Someone said, and we get this application that this one has a button and that one doesn't have the button, and then they don't call it the button, they call this one the shiny red thing. And the shiny red thing is what they're describing, but it's actually kind of the same thing. It's a button is a button, or maybe it's something new. But what is that language? And can we call something that's new and novel in our patents and then bring that back? So without this common language to describe design, we'll have a less fidelity in our descriptions. We're unable to communicate what are those design differences. We look at that to promote the sameness, that we're going to say, okay, well, because it's a button, I want to claim that it's a button when in fact it may or may not be a button. It may have changed the overall shape, the overall form, the details. So by nature, how a design relates to the task is important, and subsequently that task informs the risk of any given design, but it has to start with that design language. So, oh gosh, I'm just hitting all kinds of buttons. How do we describe design when there's no common vocabulary? How do you describe design? That's going to be in the workshop coming up, but we want to know, how do you describe that design? And lastly, another one is, how do you link design attributes to the tasks? How do you get there? So I'm going to turn this over to Megan to talk about our approach.
Thank you, Mary Beth. All right, so since we're engineers and designers interested in human factors, we are thinking about how our actions and our perceptions are involved with identifying these design features and really trying to link the design features to tasks. So we went out first to evaluate how others in design compare differences. So you've probably seen taxonomies before, maybe in biology, classifying different animals like dogs, or the Bloom's taxonomy in education. In human factors, there's some great examples of using taxonomies to classify medical or surgical complications, and also in accidents in aviation. So we set out to build our own taxonomy, trying to answer the question, can we create a repeatable method that will help us simplify and standardize classification of design attributes specific to combination products? And we think that this could be a first step in helping guide a process, a more direct process of determining minor versus other design differences. And so as we developed this taxonomy, again, we're looking to create a language that addresses all of these design attributes. And we want to look at it both from how we interact with information and how we physically interact with design attributes on a device. And so this is kind of a high-level overview of our taxonomy, the first version of our taxonomy, or one of the early versions of our taxonomy. And you can see we have categories, subcategories, and classifications that address labeling, interaction points, and feedback. And I'll kind of walk you through it a little bit. So when we're looking at different aspects of labeling or interaction points, we really went to the literature, looked at literature reviews, and tried to adopt common language from AAMI, ANSI, and HE 75 standards, and also existing FDA definitions. When we're looking at interaction points, we're trying to create classifications from everything from the way we interact with software, GUIs, inputs and outputs, what we're feeling from the device, what information we're getting from the device in a physical aspect, as well as safety features and any other physical controls associated with that device. And then to link back to our original question, can we link these design features then to tasks? We think we're on our way to a nice approach where we can create a task analysis, use our taxonomy to then evaluate across different devices. And so here you can see kind of a basic example of this, where our taxonomy cards, we've kind of sorted our different classification of different device attributes digitally across each task. So for a single task, we can compare each design attribute that's associated with each product. And in this case, we have a visual representation of where there's actual design differences. So the taxonomy, again, is identifying and classifying those design differences. The task analysis is helping us link the design attribute to the task itself. And then hopefully this would be a starting point for comparing the RLD interface to that of the proposed generic. This is our ongoing research, so we are currently actively taking comments, having people use it. and editing the taxonomy to make it more user-friendly and more appropriate for combination products. So we're interested in hearing all of your feedback today. And also, I want to again reiterate our thanks to the Office of Generic Drugs for sponsoring this research and acknowledge my colleagues, obviously Mary Beth and Molly, who are here, and then Melissa Lemke and Molly Laird.
Thank you so much. Thank you all of our speakers. Oh, Mary Beth, you can. Thank you all speakers for your fantastic presentation. And now we're gonna jump into the panel discussion. So please, all the speakers join on the stage for the panel discussion. In the meantime, I'm gonna introduce one additional panelist, David Ahern. He is the head of Device Development Center at Sandoz. He is currently joining us virtually. So, Dave, please unmute yourself whenever you add some comments or answering the question. So we'd like to kick off panel discussion from our live audience question first, and then we're gonna address some questions from the online attendees. So if you have any question, please raise your hand, then one of the staff will pass on the mic.
Hi, I had a question for Mary Beth, and this was really about the classification, or maybe it was Carrie, sorry. Maybe it was about the device classification and just trying to better understand it. So is the idea that you take that each of those are an actual design, and then like the green is a minor difference and purple is another design difference? I'm just trying to better understand how the classification is going to help categorize things as minor or other. Thank you.
I'll just use my really big mouth. How's that? So, and Megan, you can help out on this too, because so you're asking, so in those taxonomy cards, and you saw Megan had the layout of the taxonomy relative to task. So then there would be some rules, right? So visually, I can already see a difference. Is there a difference? Because I'm speaking the same language. So I visually can say, oh, this has even got 2 cards. This has got 3 cards. Or it's only got 1 card. And I can look at the colors and I can be like, okay, there is a difference right here. And then I can do a risk analysis. So the next piece of that is to do the comparative risk part of it, because the risk is going to say, is that a critical task, right? And then if I go and I apply the minor versus other, we're working on developing rules around it. So, and what are those rules? And Megan, do you remember? I don't know off the top of my head.
Yeah, so initially we thought, like you were saying, maybe if you change from a green category to a purple category, that would mean it's another difference. But as we're thinking about it, it's probably not that simplistic, right? It might be, it's another difference if it meets a certain set of criteria or some checklist. So does it require the user to interact in a different way? Like Jason mentioned before, maybe it adds a critical task. Is it associated with a critical task or a task that's associated with high risk? So it's likely, likely that the taxonomy helps us visually see where there's definite design differences and how it's associated with the task, but then you'd need to use like a checklist or some, additional human factors knowledge to say, yes, this is a minor or other difference.
Part of the question is, what do the colors mean?
Oh, that would help too. Yeah. So the colors, there's different components. So maybe one is physical controls. So that has underneath it push buttons, knobs, whatever your different options for controls. And then you can further categorize those in a subcategory. Does it have a color associated with it, a size? So each, category, maybe it's a safety feature, or maybe it's labeling on the device or in packaging. And so that's what those different colors represented, is those different types of design attributes. So they're grouped by color, and then they have categories and subcategories that help define them.
All right, this is Rob Lionberger from OGD. I have, again, a question from our academic experts while we're on there. So, I was struck by, as you think about your taxonomy, when we initiated this project, we maybe weren't thinking about this, but can you imagine using things like large language models, either to recognize descriptions or pictures, to map things on a taxonomy, right? If you think about what things like ChatGPT are doing behind the scenes, right? They have some mathematical description that groups like things together for that. So I think there's some, I think opportunities to think about linking this into, sort of more automated analysis that can also help make our review and our designs more efficient, perhaps, about recognizing that either through, analyzing pictures or analyzing text. So I think there's some opportunities to think about those type of approaches, because there is some maybe interface with
Yeah, that's a great idea.
taxonomy.
Yeah.
Love it.
Yeah, I was going to say, I think that, Rob, that goes even one layer beyond what we've thought about when this first started coming into creation. When Mary Beth and Molly were in town, we met up with them, and they had these big cards that had been printed out with like hundreds of these little, different categories and different kinds of buttons or different kinds of ways somebody might interact with a particular element of design. But the concept of putting that into some sort of a software user interface where you could actually have some sort of a software application that would allow you to do this relatively conveniently when you're doing device development or device comparison. We took it that far, but the concept of then having another back layer where you could do some sort of a forward-looking, semi-automated analysis to even before you put the human factor in would be very interesting. So we need more years and more money to keep that research going, Rob.
Any other questions?
But we have virtual attendees. Sure. So I have a question related to the ENDA guidance, which admittedly might be a very close read or a nitpicking, but question specifically about the stated intention of a CUHF study, which is to show that the generic can be substituted for the RLD without intervention from a healthcare provider and or without additional training. And I think a lot of us in industry sort of implicitly skip to the and, without additional training, without additional intervention. And I'm wondering if if there or is ever relevant, right? If there is a place and a time where you do have intervention or that can be expected or guaranteed, or additional training, how that factors into this type of process.
I'm going to come at that through the back door a little bit, Sam. So the reason we ended up having patients here at this session was because I had the opportunity to listen in on one of OGD's scheduled listening sessions with the Asthma and Allergy Network, and they had a number of organizational representatives and a number of patients who spoke very eloquently about their experience. And it was very obvious that even where we think training should be occurring out in the practice of medicine, where we think there should be knowledge that healthcare providers and prescribers know how all these things work, that they don't. And so there seems to be a big knowledge deficit amongst prescribers and a deficit of transfer of knowledge from prescribers to patients about how to use drug device combination products. I think at least I'll speak for myself and my experience and my team that we generally take the approach that the generic, you need to be able to get that generic in a paper bag at the pharmacy and take it home, and that it should be relatively intuitive what to do with it if you're experienced with the reference listed drug. We know that the way that our regulatory framework is designed, that it's always reference listed drug to generic. And we know that that does not always align with what the real world situation is, that once you have, one or more generics on the market, you have people who are being switched between different reference listed drugs because of insurance reasons and cost. We know that you have people going from the reference listed drug to the generic. We know that there are people switching from generic to generic, and that very often individual patients have very little, if anything, to say about it, that there's contracts and pharmacy benefits managers and all sorts of things going on in the background that take those decisions out of patients' hands as well as out of prescribers' hands. So our general approach has been that our entire regulatory framework basically says you need to be able to hand this to the patient at the pharmacy and have them be able to figure it out and be as likely to use it correctly as they would that reference listed drug that we're assuming they have previous experience with, because that's what our regulations say is our bar. That's our bar for approval. I'm trying to think of a situation where you could have one or the other, but I can't think of one, and I think that might be just a prepositional thing, or can you think of a situation where it's one or the other? Oh, good. Irene, did you have something to add? We need Irene up here.
I was going to say that in principle, right, if there is, let's say, a shared system REM type of situation, or that kind of like an administrative control that might be in place, that could be a scenario where you do in fact have some sort of intervention that's built into the system surrounding the drug, right? Because that's a risk management program. So, I mean, that's not necessarily a common situation, but it's something to be aware of.
I was just going to suggest that for in the new drug environment, when people first get those drugs, there's often a learning curve. And when we have a generic, we're making this assumption that folks have prior knowledge or prior use of the new drug, but that's not of the original reference drug. But that's not always the case, because as generics take up more and more of our prescription drug ecospace, we're going to have a lot more patients go right to the generic drugs. In fact, I think that's generally the case, that patients sometimes get the generic drug as their first drug that they get. Then so there's that learning curve there in that space, and then they may have a reverse issue of not being able to use the reference drug appropriately if they get that substituted at the pharmacy. So I think it's an interesting space, and maybe there is something to be said for having universal training for all these devices, and that would lower the hurdle for the changeover too, potentially. People learn to do a lot of things on YouTube, on social media, things like that. So there is a place for training in the virtual space without having a live trainer. I think there are other approaches to having good labeling, good training, and I think we may want to get into the 21st century on some of those training aspects. Thanks.
Can I add a comment?
I maybe make a comment, not as a CRCG director, but so my son has type 1 diabetes. He was moved from Admelog to Lispro, just formulary change, right? We go in, we fill in prescription, nobody check it. We get it in a brown bag. We do not know until we get home that there is a different name and look different. So the kid smart kid, he Googled that at least it's the same drug, right? Nothing completely different, but there was a small change in how is dose, like how you measure exact amount that you get dose. So, I mean, smart kid, he could figure out, adjust it. I don't think the generic version, I think there are two different drugs. It probably wouldn't meet your criteria of fully substitutable, this little difference. However, as a mother, relative to not being able, don't having access to cheap alternative to insulin, this difference is really minor. And this is, I think, where most of generic people lay. And here I am, the center director, but also a user. So, and this is my opinion.
Yeah, and I, one of my favorite slides I've seen today was one that Johannes put up with the scale. And I know that as long as I've been working in the drug device combination product space, which is fascinating every day, it really is all about trying to walk on top of the fence without falling off. And you're really trying to balance getting that same clinical effect and safety profile and achieving access. And, it's a really interesting mental exercise to think about, well, if I was asked to write what our regulations should look like now, today in 2024, instead of 1984, what might I do differently? Because the whole landscape of drug products and drug device combination products looks so different now than it did 40 years ago. And for anyone who's talked to me about it, I'm a huge health education person, and I'm always thinking about, education and what people know and how to make patients better able to manage their own conditions and be active members of their care team. And there are definitely holes there. We know that people access information differently. We know that people learn differently. Some people may learn really well from watching a video. Some people may need to hear it, and have somebody right in front of them. Somebody may just prefer to read it. People process information differently, and people aren't all equally careful about where their information comes from. So having two sons who are in their early 20s, and when I have one of them come tell me something, I'm like, well, where did you hear that? Or where did you find that out? And sometimes they can't even tell me. And so we know that different sources of information have different levels of reliability and different levels of truth in them. And I think it's a challenge that all of us need to think about, because yes, we're regulators, and most of you are doing design and development and running your businesses and trying to keep them financially intact. But the mission is to really get these products to patients and help them take good care of themselves. And there's, I think, a huge educational information sharing and understanding deficit that I think only adds to this problem and makes it harder for us.
So I know we want to get to a new question, but just to get back to the original question for this great dialogue. So yes, the additional training and intervention without intervention of a healthcare professional. So a few points to make. One, I agree, but we have the understanding, what we've learned is that there is not going to be any intervention. There's unlikely to be training. And that's for us when it's being actually substituted, or if it's they've only received the generic. But even if they're only receiving the generic, the whole basis of that approval is still comparative to that RLD, where it's still that same expectation that if you got the RLD or the generic, you wouldn't need, they would have the same clinical effect and safety. You wouldn't need training for either scenario. But there are scenarios that do exist, like the REMS is perfect. We haven't seen that in the generic user interface space, because a lot of these products that are REMS that are regulated as drugs and generic drugs, they just, very few of them, if any of them, they're not drug device combination products, and these user interface questions just haven't come up. I know there may be some biologics in the interchangeable biologic space. This may be in the future, but we just have to recognize that the vast majority of these cases, the point of access to these drugs for the patients is, here's your bag. It may be you may have taken it before, it may be different, but the expectation when there's that comparison back to that RLD product is that whichever one you get, either one should have the same clinical effect or safety.
So we do have a comment from our panelist online, Dr. Dave Ahern. Dave, go ahead.
Great. Hopefully you can hear me. Maybe I'll just check that first.
Yeah, we can hear you.
Great. Thank you for allowing me to join virtually. I'm unfortunately traveling in Europe this week. First of all, great presentation so far, and I'm really enjoying the discussion. I think what has really resonated with me was Carrie's presentation, where we talk about kind of state of the art versus just copying what the oral D has. And I think there's an imperative as a medical device or drug device combination product developer to really focus on safety and efficacy and usability. And then a layer on top of that is substitutability. So I think it's certainly at Sandoz, what we try to do is to ensure that we're not using substitutability as the only means of validation for our devices. I think you still have to have a robust design and development process, human factors process, and verification and validation process to make sure that the device is appropriate. And then on top of that, if you have the freedom to design in such a way as to make the user interface, excuse me, as similar to the reference product as you can, that's great. But where it becomes a little bit tricky is if you know that there are known use errors with the existing product, and I could give some examples, but it may be not appropriate. Sometimes the feeling is that, well, the reference product is out there, even if it has use errors, we shouldn't try and improve upon that, just copy it, right? That's the kind of the default position. And I just wondered if the panel had any thoughts on where we can identify means of making a device more intuitive, more safe to use, simpler. Should we be striving in that direction, whilst at the same time keeping an eye on this whole topic of direct substitutability? And again, the point that was made earlier about that, not every patient that gets a generic device is trained on the reference product. They may be getting the device for the first time. So yeah, just an open question to the panel.
So is this working? Can you hear me?
Mhm.
So I don't want to send a message, certainly, at least from our office, that we're against innovation and we're against making improvements and things like that. I think, though, that the regulatory standard is substitutability, right? And so there are mechanisms in place by which you can do both. Whether that indicates that I need to have some comparative use data or some other data to demonstrate that substitutability can be met, that's the standard for, right? That's the standard for a generic. So I'm interested in what my other panel members have to say.
Well, I'll add that I'm in strong support that we improve it, as a human factors engineer and as a designer. I don't think any design team, any product development team, and we were talking about it earlier, there's no design team that wants to do a repeat of bad things or just copy paste something. It's not an exciting thing to do. It's not fun to do your job at that point in time, and you're not actually making an improvement. And I think one of the goals of the taxonomy is to point out that, in that process of having the common language in order to compare two different devices, we can actually say at that point in time, this device is a lower risk, and have confidence from a regulatory perspective that I have a level measure. So I am using the same equipment to measure these two designs, whereas right now we're relying on narrative and relying on descriptions that are highly variable when you go to the other versus the minor, and the propensity and the desire, even in the question, we heard it too in our interviews at the start of the process, where there's you want to be minor or no change, because that's an easier argument in your submission. But having something that can level it and to communicate it in a way that these new improved devices, where I have taken those known use errors and I have improved the design, is actually a lower risk, I think will go a long way. And to your point about simple, we have to get more simple. Having any combination product where a user group that is for an elderly person that requires 25 steps is absurd. I mean, I don't want to do 25 steps, and I'm not considered that old. So I think we have to get simple. I mean, I'm old, I'm not that old.
Okay. So we have a time limit, so we're gonna take one or two questions from the online.
Shinae.
I think this question ties back to using or a generic DDCP referencing a discontinued RLD. The question is, there is no more RLD available in the market. There is no question of negative transfer. Why is a comparative analysis still needed? Why do we need a comparative analysis in that case to be compared against the discontinued RLD?
So in that situation, the reference listed drug is still, it's the flag bearer of being the standard, because when it's not available, if you don't have a standard against which to compare, you're going to have the wild, wild west. If you're having multiple products in development by multiple companies at the same time who aren't talking to one another. And so even if it's been discontinued, as long as it was not for reasons of safety or effectiveness, that reference listed drug provides a standard against which all of those other products are going to develop, so that when they are substituted for one another, they are going to be within that therapeutic equivalent substitutability range that we're seeking. And that is the reason why it still goes back to the reference listed drug. Does anybody have something else? to
Yeah,
add to
I
that?
agree. And then it's also, as the presentation discussed, like, if they're all discontinued, like the traditional comparative analyses is kind of, you're handcuffed. But that's why we incorporate other information about additional supportive, whether approved ANDAs or other ANDAs. Getting back to the naloxone example, they were able to compare their product to the RLD, but also in the context of what else was on the market and like how in real world that would be quote unquote substitutable, because that's what, the end user may be used to. So, but again, that's the standard.
Thank you.
Thank you. We have on the following session. So this concludes our panel discussion. Thank you so much for all the participants and speakers. Now I'm going to move handing over to the podium to the Robert Lionberger for the next session, user interface time with FDA Q&A session. Thank you.
So I'd like to invite our panelists up to the podium. So we have Howard Chazin, who's the director of our clinical safety and division of clinical safety and surveillance in FDA's Office of Generic Drugs. We have William Chong, who's the director of the Office of Safety and Clinical Evaluation in the Office of Generic Drugs. We have Jason Flint, who's been on the panel before. So Jason, you have to come back up. Is the deputy director in DMEPA in FDA's office, CDER's Office of Safety and Epidemiology. We have Stella Grosser, who's the director of the Division of Biostatistics 8 in the Office of Biostatistics. And we have Markham Luke, who's the director of the Division of Therapeutic Performance 1 in my office. So welcome to all of our panels. So this, is your question time. I think, people always try to ask FDA questions at all of the other panels. Right. And so probably should have banned the FDA staff from the other panels so you can ask your FDA questions here. And so I think this is a fantastic opportunity to have a, wide-ranging fundamental discussion of the issues related to combination products. So I encourage questions forward, or else you'll have to ask, or they'll have to answer my questions, and they probably won't like that because it'll be very challenging and controversial and generate lots of problems. So please ask them some questions so they don't blame me for the hard questions.
So I might actually jump in, not with a question, but with a comment. I think there was a slide or two earlier today. I just wanted to point out that when we talk about critical tasks, there are actually two definitions for critical task. One of them is for CDRH. And so if you see the word severity of harm or severe harm, that is the CDRH definition for devices. What we should be talking about here is the combination product critical task definition, which does not include severe. So we're looking at just harm. So I just wanted to clarify that point. Thanks.
I have a procedural question about the HS study protocols. When they're submitted as part of, are you able to hear me?
No.
Microphone to the. Shoot.
Yeah, I think you have to speak up a little bit loud, like hold the microphone close to your mouth. The microphone's on, so just close and loud.
So this is a procedural question about the HS study protocol. When it is submitted as part of a CC or a post CR with a protocol, we are recommended to include some questions on the protocol. So it's possible that sometimes we may not realize that we are not asking a particular question that is actually of concern. So does agency look into that and provide and feedback, even if we've not specifically asked a particular aspect of the study?
So, it depends on what that is. So one thing, as we noted, when we get a protocol, we're not going to read through the whole protocol and then give you comments on every little bit of the protocol. It behooves you to have sufficient expertise as the developer, and applicant, to put together a protocol, to have the right clinical, right study parameters, et cetera. If you have specific questions about the protocol, that's where we ask you to come up with a list of those kinds of questions that we can answer together with our colleagues in DMEPA and DCR. We have conversations about those specific questions. As far as missing out on like questions in your protocol, I think that's an important aspect to have that kind of expertise, and hopefully you're able to hire that kind of expertise for your company or through a respective contract research organization to get that expertise in the drug development expert ecosystem. Thank you.
Yeah, I think we really prefer you to ask questions about the protocol, to focus our attention on what you're uncertain of, right? We don't want the protocol business to be like, we're going to check everything that you did, right? And you don't have to put, we really want you to identify the questions that you have. And then I think it's, that also allows us to get the questions to you on a faster timeline, right? I think you can ask questions about a protocol through our control correspondence process, which is our fastest way that we can answer questions, right? And so if we set the bar as we review everything you sent us, then that's, we're going to come up with longer timeframes for those.
Right. Through our GDUFA commitment letter, we committed to answering those kinds of questions, but we didn't commit to being the consultants for the development program. Thank you.
Yes. Is there an effort or anticipating an effort at FDA to harmonize terms and definitions between CDER and CDRH? Because some terms in CDER have different meaning in CDRH. I mean, like you just mentioned on critical tasks.
So in the context of human factors, that is the only term that I'm aware of. If you have others, I'd like to hear them.
Oh, one is validation.
Maybe we can talk offline.
Yeah.
Like,
okay.
So I'm curious if we're talking about difference in the term validation. You're talking human factors. So there's multiple types of validation.
Exactly.
Right? So when I'm talking validation, I'm talking human factors validation. That's my domain, right? But there's certainly sorry. There could be design design validation, right? As well, and that's certainly more a CDRH type of term. When we talk about, at least in DMEPA and in OSE, we talk about validation. It's typically human factors validation, right? So there's multiple definitions, but I wouldn't focus on the term validation. I would focus on what validation are you talking about?
Yeah, it's the context in which it's used.
Correct.
I would say there's very minimal discrepancies in understanding of the language. Some of the terms might be unique to, say, devices, but we have CDRH devices, former CDRH reviewers in OGD as well. So there's a lot of crosstalk between our groups, and we do have discussions with each other. Thank you. Yeah.
Also, can I ask another? Something a little different. We were talking about not designing in known user flaws when you're designing new devices, but you really can't put those new or those less the designs that that result in less user error as a claim in your label, because the label has to follow RLD. So is there an incentive to designing out those when you can't claim anything?
Well, could I ask a clarification about that? How would you know? Because I think there's a lot of assumptions that are made, and I'm on the safety side. A lot of post-market safety issues are, I think the best efforts go into some new device drug combinations. And then when they're out in the market, we suddenly get these reports that's not intuitive, or people are confused, or the strength they need to maybe open something or is not, was not considered maybe in the population. Maybe it's a neural population with weak hands or something, or a person who can't see very clearly. So I think that that a lot of this comes from a life cycle aspect, and that we're still learning, I think, because it's one direction, because I think we always started with the RLD, right? And that became the innovator product. And then generics started, but now we're a little further down the road where we have multiple generics on, and everyone kind of takes a different viewpoint of this. And then that's the problem, I think, is the cross usage of these products.
Yep.
And I think we're going to talk about manufacturing. Were you going to go, Bill? Sorry.
Yeah, you can go ahead. I'll wait.
Okay. So an evolution of design changes, there are numerous reasons. There's the discussion of safety for a product, problems with manufacturability, problems with the device failures that could lead to certain changes. And we're going to talk a little bit about manufacturing stuff tomorrow. But the whole idea of having a device that's easy to manufacture, doesn't go wrong as you do your sterile fills, et cetera. That's going to be really important in the context of having a product that you can make and put out in the marketplace with minimal fuss and minimal recalls. Thank you.
Yeah, I was just going to acknowledge that. I think, in the generic space, there are constraints on what sort of labeling, what sort of things you can put into language. And I recognize that from a business perspective, it really handcuffs you in being able to put a marketing edge on your product versus somebody else. But unfortunately, that's sort of the situation we're left in. I think in terms of if that's something that you want in your development program, a 505(b)(2) might give you that avenue to be able to demonstrate that you have an advantage over another product. And I think, and this may be less valuable to you as an incentive, but if your product works well and works better, theoretically, you will get fewer complaints, and potentially the ability to get fewer complaints from that may be useful in terms of just your post-marketing surveillance and your safety, the costs that go into monitoring the safety of your product.
Yeah, I mean, the marketplace also should maybe it's unrealistic to say that there's a marketplace for pharmaceutical products, but in a free market, right, people will compete on improvements, and if it's easier to use, you get good reviews, and you get and there'd be some market mechanism to encourage more usability on products as well. But I'm not sure that you and the product developers can tell us whether that's the reality or not, or it's driven by other things.
I have a question. So is there a defined time frame for the protocol review for this comparative use human factor studies?
I think your question is if you submit a question to FDA, when you'll get a response from it on a COHF protocol?
So typically we used to submit protocols itself, but now I'm trying to understand, and then the time frame for the review or any comments from the agency were fairly extended. So I'm trying to understand, procedurally speaking, should we be even submitting the CUHF protocols, or as earlier you guys referred, should we just provide you the gist of the protocol? That way we'll get faster responses.
So I mean, I think for the generic program, we have a user fee agreement, right, which has very specific timelines. And as all of you here on the panel know, we're really serious about meeting those goals, far exceeding the 90% metric. So, the timeline will be dependent on the user fee context in which you ask the particular question, right? Whether it's a control correspondence, 120-day answer, or it's in the context of a meeting, right? So that's one aspect of it that you'll that's how long it will take to get your answer. And that's why I think the earlier discussion around, don't send in a question that says, can you read my protocol and tell me if there's anything wrong, right? That's not really what we're after. But it's the idea is that, depending on the context, right? Sometimes it's very helpful to send in the whole protocol and here are my questions. So then the reviewer has the context so they can give you good answers to your questions. But the time frame for getting those answers to the questions is driven by the user fee agreements around the GDUFA program. So if you're talking about, new drug development,
this is for ANDAs itself,
Yeah,
for generic drugs only.
Yes, for generic.
And
So
when
follow
we
these-
submit the CUHF study protocols, I have experiences where the review of the or any comments on that were not received for many months. So that's why I was asking, what should we expect?
So I think the first question I would have is, was that under GDUFA 2 or GDUFA 3? And under GDUFA 3, we do have timelines for responding to controls, including for questions related to CUHF protocols. As Dr. Lionberger noted, like the question is not, here's my protocol, can you read it? The answer is yes, I can read it, but I don't know that I will be giving you any useful advice without understanding what your questions are. And so when you have questions, we're going to be targeting those GDUFA timelines that are in the commitment letter to address those questions. If it's a question in CUHF protocol and a control, it's 120 days, and I forget what it is in a pre-ANDA, but so I think that's it's also 120 days.
It depends.
Great.
I mean,
So easy.
again, so now under GDUFA 3, you also have, perhaps the question is, previously, if you had a question about an application that was under review and you wanted to send us a question, there was a sort of a way that things could go into a little bit of a black hole. But now under GDUFA 3, you have the opportunity to use the control correspondence process after you received a CR to answer questions. If you're sort of like, you've got deficiencies and you're developing a protocol in response to those, you have opportunities also through the post-CR scientific meeting to have a meeting. In both of those, it's appropriate to send in the protocol as part of that meeting package and the questions, or send in questions about your protocol through the control correspondence process. So that's something that's new in GDUFA 3. If you're in the context, previously under GDUFA 2, those were available before you submitted the application. Now you can do them, when you're in a CR status as well.
And I would stress again, like and how Jason and Stella back me up on this, that that we see a variety of quality in the protocols coming in for our studies, and it behooves industry to think of to make sure that you've adequately invested in an appropriate consultant to develop your protocol, that you don't just go to a fly-by-night CRO or someone who's doing this for the first time maybe, and they're not sure about what they're putting together. So do think through who you're talking to. Do they have experience in? We've had some come to us and say, okay, well, I've developed a whole bunch of protocols for OND, for new drug development, but the whole idea of what do you do for a generic, that was totally new to them. They didn't understand bioequivalence. They didn't understand the CUHF paradigm. That should be a red flag for you in generic drug industry. Thank you. And Stella, if you want to comment on good statistical practice or something, since she's on
The person here with them.
Sure. The setup for equivalence in the traditional frequentist approach, or is it, there we go. So I was just saying that equivalence kind of twists the order, if you like, of the statistical hypotheses that are tested. And for people, I see this within my own office, who have only worked in new drugs in a superiority kind of proving ground. It's a very different paradigm is too strong a word, but it's a different way of thinking. Yeah. And I also want to say that I was noticing the map that was mentioned earlier this morning about who's doing which piece of the application. And our office was not quite mentioned, but I wanted to let everyone know the statisticians in my group are there. We're putting in our 2 cents trying to keep everyone, I don't want to say honest, but including within our panel here. So thank you.
So I have a question about something I know people are struggling with in this comparative process. And the ANDA guidance requirement that the device and the labeling be the same. One of the problems we see with combination products that are going off patent and are now ripe for generics is that they were developed 20 years ago before human factors really became well-known, well understood, well practiced. And so we have a lot of old products with terrible instructions. And the ANDA guidance says that our new instructions have to be identical. And I would just like to make the point that I believe FDA would accept new instructions that are better than the old ones, particularly if it helps people to use both the RLD and the generic. And I also suggest that that would be a great thing to submit along with your protocol. Does that make sense to you?
I'm going to start with this one on the safety side. Okay, so yeah, that sounds like a grand idea. And what I wish would happen is that there would be standards that would come across, like, for example, standards for syringe markings, for example. When you start seeing, I know, but they're not in the products, it's the older products. It's either the barrel or the syringe. Here's the issue is you're right, the RLDs are old, but we're constrained by the RLD's language. And I'll tell you, a lot of companies do come in and try to improve the IFU, and then we get these reviews, and then people argue, and we have lots of internal meetings. I'll just say that if you talk to enough patients, they're not reading the labeling in the new IFU. It needs to be, intuitive. You pick this thing up, like they were saying before, or Katherine was saying before, but you know what people do. They get it from the pharmacy, they unwrap it, they want to use it, and no one's reading this long instruction sheet or micro printing. They're not even seeing it. So I think that that's a thing that's a grand idea. And I wish the industry would do that, would go back and say, hey, RLD, can you update? Can you become modern? Because you should all work together. I'm serious.
So Howard, it's a great idea. And tomorrow we're actually going to have a discussion about standards.
Terrific. So
I still have a comment. I want to hear.
Yeah, go ahead. I'm just
there's some
wonderful. So, I think from my perspective, certainly we do want to see that, if the 30-year-old RLD has a problem and you fix it in your generic version, then yes, the labeling should accurately reflect how your product is used. But it does come back to, one, the product. We need to get to the point where we think that the differences are okay. And if we're there, then we can start having a dialogue of like, what are acceptable differences in labeling under certain, the regulations say there are certain exceptions which can allow differences in labeling. So if we can get to the point where we think your product can be substituted, it is okay, we're fine with it as approval under an ANDA, then we can start thinking about like, okay, are these labeling differences something that are acceptable under the exceptions that we have in our regulations?
So yeah, I think that's the important point of what Bill is saying. Is if you read our guidance carefully, it talks about sort of hints at what types of labeling difference are acceptable. That's really predicated on, first, as Bill said, FDA deciding that the devices are actually substitutable, and then the labeling reflects the device that FDA has said is substitutable. So it's it's a little bit subtle difference, and and it is, I think, certainly agree with the sense that it can be very confusing with saying like labeling has to be the same, except for the places where it doesn't have to be the same.
Exactly.
But I think that guidance does give some hints to the direction about thinking about the evaluation of the product first, and then is the labeling appropriate to the product that is acceptable. And I think the way, that's an important aspect of thinking about what types of flexibility there are within that type of system.
So on Howard's point about standards, we are going to have tomorrow a discussion about standards, and those ISO standards, those are industry and FDA code-driven standards. And within those standards, you actually have the ability to guide labeling for FDA. And so since all of FDA is co-signatory to ISO, then you can, as a generic company, point out that we want to have our labeling meet the current standards, and the CEDAR, you should use the current ISO standards for labeling for this auto injector or this prefilled syringe, et cetera. And so then, and that way you can change labeling that is years old because you're saying this is our product meets the current ISO standards, or I don't see many generic companies on those ISO working groups. I don't see many drug companies on ISO working groups, but we send in CDRH, we send folks to those ISO working groups to make sure that these drug delivery devices meet the current standards and are safe and effective and accurate for use in delivering drug. So I think it might be interesting to make sure that generics, you do have to pay to be on these standards working groups, but I think it could be a useful investment for generic industry to think about. Maybe an AM might be interested in it.
So question from me is that whether FDA has any plan to make the consistent terminology for different dosage form. For example, Wegovy and Ozempic, they are launched in US market, but the one is in auto-injector form and another is a pen injector, it's a multi-dose. But the terminology of the product is a pre-filled pen, both. But that might give some kind of confusion to the end user. And some of the pre-filled syringes, they have the auto-injector form, but their product description says it's a PFS. Some of the cartridges, they have terminology as a syringe on the product labeling. So my question is that, is there any plan from FDA to make it consistent terminology across the other products or different dosage form?
I would just say that, I think FDA recognizes that nomenclature of dosage forms is very difficult and governed by lots of historical and other aspects to that. And, I think certainly people at FDA would hope for consistency, but I think some of the challenges really are that you have 50 years of drug products with names that are established. And, so there's lots of difficulties and competing interests in any kind of nomenclature normalization. But there are standards committee. Mainly, I don't think we have anyone from OPQ here. Product Quality Office is generally the one that's responsible for the product nomenclature. And certainly there's lots of ways they work with USP in different groups and CEDAR data standards and nomenclature. So there are efforts around normalizing these things, but it does carry the weight of, traditional pharmaceutical practice for hundreds of years in that. So I wouldn't be optimistic that FDA is just going to tell you the difference between a pen injector and an auto injector tomorrow exactly. But I think, we do try to clarify that, as many of these things as possible going forward. We're recognizing that that older products are more challenging as well. Mm-hmm. Yeah, but I would say this personal thinking is really the comparative analysis. You ought to sort of elide that, what did I call it? And really focus on the steps that are in our guidance and what does it actually do? And I think that's the important way to think about it is, as you go through those steps, are those steps the same rather than, they called it a X and I want to call my product a Y because that's the current standard. Really go through the steps of the analysis because that's the critical thing. If you're, you want to be the same as your reference product in those matters. And so, and then the labeling issues and what you call it, that's something that, sometimes you have to call something that's not optimal because of the sameness requirements in labeling. But it's really the comparative analysis that's the important thing that you should focus on rather than getting confused by I want to call, this labeling calls it something that isn't the current, favored term.
Yeah, and I just want to throw my hat on that in that ring. Exactly correct. I think terminology is one thing, but I'm looking at two devices side by side. What are the differences? How do those impact tasks? How do those impact critical tasks? How does that impact labeling, et cetera, right? And that's what we're really focused on, right? As opposed to pre-filled pen or auto injector.
I have a question about terminology again. So regarding design validation, which we talked about a little bit earlier. So the intent of design validation is to validate the final design against user needs and intended use. In the combination product world, there's so much focus on the human factor study or validation. Do you see anything, is a human factor study or validation testing is sufficient, or you see not necessarily that's sufficient. Because, typically in the device world, we have very explicit traceability between every user need to the validation that, human factor testing is one kind of validation. There could be one other means. How in the combination product field space, FDA practice that design validation?
So your design validation is going to be multifold, right? You're going to have the device in hand. You're going to see if it works properly. You're going to see if it compares adequately with the reference drug for the generic space. You're also going to validate it for manufacturability. Does it come out of the manufacturing process intact and usable, and especially when you scale up? And also for injectable products, you're interested in sterility, and some products have problems with sterility after you manufacture it. So and shelf life and all of that. So we have two offices that look into that in OPQ, and they review it, and they also test those products. So I think that's additional product validation that I think we're going to talk about tomorrow as well.
So just adding to a little bit background for example, a general infusion pump, which is a delivery device that can infuse hundreds of different medication used in the hospital. So to the end patient or user, they're one kind of drug delivery combination product that they generally can be used for multiple medication. So then when we do design validation, we not only do human factor study, but there's also similarly testing, run every step what a user manual typically provides, and trace to the individual user need. So make sure every user need defined is validated. So how the combination product side actually is your focus really on just the human factor, or actually you do look at the broadness of design validation as well?
So let me just make sure I understand the question. So it's both related to the usability, human factors aspect, but then also separately the device performance and robustness, that sort of question. Like, are we looking at both, or are we just looking at one? Is that
right?
The question?
Basically, look at every user need. However, because user needs are one of the key document defined at the beginning and in the end need to be validated with the final design and see whether that needs is adequately met. Yeah. So that's the concept.
Okay.
So human factor study in that context is really one kind of the validation, because your human factor study can have maybe your test are not defined correctly. So in the end, you may not necessarily achieve the intended use or user need. So I just don't know how combination product division practice that terminology.
So I'm going to look over here a little bit just to make sure I'm not saying anything completely wrong. So I think when we're looking at combination products, they have to both address drug regulations and device regulations. So the device regulations and the quality assessments will include some of those things, I think, that you were mentioning today. And I think that's the focus of tomorrow's symposium, is really looking more at the quality aspects of combination products. So hopefully that was answered.
Yeah, I think a lot of the, I think here today, most of our discussion is focused on the user, because the user is a significant variable that's not under control. But there's a lot of aspects of device performance, right? That's objectively measurable through laboratory testing and performance, But I think generally that's, just part of it's how FDA reviews it, and through the product quality review, through the engagement of our CDRH colleagues in the review of the products, that those aspects are generally not here we're focusing on things that are related to the users. So down front here.
Maybe since we have you put yourself on the spot, maybe I can ask a policy question. So now we have been talking about how we show substitutability. So let's take the situation. We have the RLD approved and maybe 1, 2, 3 generic products approved as substitutable ones. Is FDA in any way evaluating then, since we've heard from the patients that they are being switched also from generic to generic, substitutability between generics? I mean, there might be like a minor difference from one generic to the RLD and a minor difference from the other generic to the RLD, but the comparison between two generics might show that there are like non-minor differences that are not addressed by any data. Is this something that FDA is considering? And another question, once substitutability has been shown, what happens if the RLD wants to implement a design change? Would the sponsor of the RLD be requested to show then substitutability of the changed design to the original RLD so that the generic, as the therapeutic equivalence rating to the generics remains valid? Is this something that FDA is considering or doing?
If I can answer if you don't. All right, so I think those are very interesting questions. I think let's talk about the first one. So generally FDA is thinking on, well, what if people are switched from generic to generic? Our general thinking on that is that's why we designate, if you think about for bioequivalence, we designate, if the RLD is not available, we designate a reference standard. Everybody compares to the common reference center point, right? And so that's why you heard today, always, trace back to the RLD, trace back to the RLD. That's sort of the control over everyone, staying sort of close together. But, I think in general, I think in most cases, my sort of feeling is that's often, more of a hypothetical problem that, a generic will be, more different than another generic, and then you'll have a bigger difference. People have talked about that, but there's very little evidence that that actually is any kind of significant problem. But in general, the answer to that is the centering on the reference product. So that's your first question. Second question, also a challenging one. And this is not just limited to drug device combination products. Any generic product has some risk, some challenges if the reference product makes a change after there are generics approved. And that's a very, and I think all of my OGD colleagues know that that's a very complicated situation. We try through various ways internally to be aware and coordinate these activities to minimize the disruption. But occasionally, if the RLD makes a change, then future generics have to match the current RLD is generally the thinking around that. But like you said, what if there are generics on the market? Do they have to go back and change that? The answer is sometime. I mean, in reality, mostly not, because generally the RLDs aren't making such significant changes that they're not bioequivalent, substitutable to their previous version. If they're making a really big change, sometimes then it becomes a new product, right? So there's some controls on the, you can't make any kind of change you want in the reference product without reason or labeling or consistency. So it's not a free for all, but it is possible. And those cases are very, challenging to the process. And, certainly there are cases where, especially if that change is to the RLD is motivated by safety, and that change then has to propagate to all approved generic products. And there are mechanisms for FDA to chase those down and make them in cases where that. But in reality, there are cases where the RLD changes, FDA asks the new generics to match the current RLD. But if there's not, Howard can comment on, if there's not reports of safety signals with those generics that are on the market, there may not be a need for them to make the exact same change. But there are a lot of subtleties to that point. And it is a challenging part of our system. So,
and there have been instances where with the RLD change, they need to send in clinical data to support that change as well.
And I want to say too, once a number of generics get on the market, there tend to be market leaders over time. So you don't get like 10, 15 generics on the market at the same time. You usually get the top 3 or 4. So it's market factors that drive some of this. And in a good way, we're not seeing a lot of problems when we're seeing, maybe there's an occasional generic that has a problem. But I'll say for the most part, when in the life cycle, while most generics are doing just fine, product combination products, we have only had a rare set of cases, and we handle them case by case. So I will say we're doing a pretty good job out there. So I want to say something.
I'm just going to assume you don't want to start doing that. Is that a fair guess? Because I mean, just because I think
the second part
on both parts, right? Like to start asking that you compare to every approved version of the product, and I think that would be extremely difficult for you, I'm assuming. And to Rob's point, I think certainly the anchor of the RLD is something that I think we expect will prevent that from being an issue or a challenge or a problem as we approve additional generics.
And there have also been instances where the RLD would come up with a new iteration of their product and then abruptly recall that product because they had failed to consider certain aspects of manufacturing their product. And I could think of 2 products specifically, but I'm not going to say what they are.
Good idea.
I've got a simple question. This ANDA guidance came out in 2017. It's been 7 years. And ever since there have been lots of questions and different interpretations. And I'm sure the FDA is learning as well as we are learning. So when do we expect the full guidance to come out and whether this usability guidance will be more usable next time?
So I'd say the only answer you can get to that is FDA publishes a guidance agenda. And so if we were going to do something, go look at the guidance agenda. But other than that, generally, we're not going to talk about when changes to guidances, come out other than that sort of process. So,
and the link is in Lisa Burcue's talk.
So I would have a question with regards to kind of a global human factors approach. So in Europe, adherence to the human factors IEC standard 62366 usually involves a summative human factors study. We've had some thinking at Sando that we usually are developing a global product, so we're performing a summative study, and some of these summative results could be supportive for our human factors evaluation in general. Is there a situation that the FDA could imagine where some of these summative results, in addition to supporting the outcome of a threshold analysis, might also be enough to prevent the need for a comparative use human factors study? For instance, in justifying why a certain difference would be minor rather than other than minor?
Depends on the device.
I'm going to start by making a pitch for Symposium 3. Where we do want to have those sort of discussions. I think the answer, and I always joke about this, and people that know me know that I joke a lot, but that my favorite and most scientific answer is it depends. And so, right, it's in the details, I think. And so I would need to know more about that type of approach and those types of things. But I think those are things that we want to talk about in Symposium 3. And so I'll just leave it there. Thanks.
So I think that means we've come to the end of this panel. So first, I want to thank all of our panel members for the great discussion and the questions that you brought up. And appreciate that. So I think the panel can be dismissed, and then I will have some closing remarks for the online session. So you guys can brief this problem. All right. So again, if you're here in person, this is not the end of the meeting for today. We will be having our breakout sessions and small group discussions. However, if you're attending online, this is the end of today's session. We welcome you back online tomorrow at, give me a time, something like 9:45. Okay, and for the continuation of the live stream session. But it's been a very fantastic meeting. I really appreciate the organizers for the work. This has been a spectacularly successful program. I think, everyone here has learned a lot. Hopefully all of you online have learned a lot. Want to give you a sense of how good this workshop was. So during the workshop, my wife, who happens to work for FDA, texted me a message. She said, "Great workshop. It's really useful for reviewers to know the patient listening session was awesome." And just a hint, been to many of these workshops, never sent me a text like that before. So, at least that FDA audience is very happy. And I think it's very, I think that's very true. We got a great set of perspectives here today, and we look forward to having you here tomorrow to continue this activity, but also but for those of you in the room, we have, again, some great in-person interactions here as well. And that's one of the things that we're trying to do with the CRGC workshops is have some things that you can't really do streaming online, have some really open face-to-face discussions with people from a variety of different perspectives, and again, learn from that. So again, as you think going forward, really consider if you're really active in one of these fields that the CRGC is having the workshop, to consider the in-person attendance here, because there's some fantastic in-person attendance, in-person activities that you can't capture online. So with that, thank you all for attending, and remind you to come back here tomorrow at 9:45 online. Thank you all.